In this series of modules, I will talk about energy and how enzymes, remarkable workers in biology, can manipulate the system and perform miraculous reactions. Let me give you an example of this. Nitrogen fixation. On the top part of the slide, we have a molecule of nitrogen with 3 molecules of hydrogen, resulting in 2 molecules of ammonia. This is the Gerber process, done industrially to produce ammonia. Let's look at the conditions that are required to conduct this reaction. This reaction requires 500 degrees Celsius temperature and a pressure of 150 to 250 atmospheres. That's a huge amount of pressure, huge amount of energy required to simply form ammonia from atmospheric nitrogen and having it react with hydrogen atom, hydrogen molecule. So this reaction occurs in nature also. This reaction combining nitrogen with hydrogen happens in the roots of certain plants. These plants have special type of bacteria that make a remarkable compound called nitrogenase. This nitrogenase, the enzyme, can perform the same experiment in ambient temperature, whatever the temperature is outside. It can take a molecule of nitrogen, combine it with hydrogen, and produce ammonia. So there is a stark difference, an example of the power of enzymes. They can conduct reactions which have requirement of large amounts of energy. They can conduct those reactions with a very small amount of energy. So it wouldn't be wrong if we say one of the biggest miracles of life is that it can perform slow reactions and the ones requiring extreme conditions quickly at physiological conditions. Physiological conditions are the conditions in which we exist, or our internal environment. In order to understand how enzymes work, we have to first look at certain rules about energy. So I'll talk about energy in my next module.